I apologize for technical difficulties there. My PowerPoint program seems to have uh, crashed. And so this was the third time I was working on this video. I decided to not start over and just stop and give you another short one. So anyway, I apologize for that and all of the other technical difficulties that I have with this thing. All right, so let's talk about what Meselson and Stahl would have seen in their experiment for each of these types of replication. So in conservative replication, if we start with our N15 DNA, and then we transfer our bacteria into the N14 DNA, we're going to end up, because we are conserving the original molecule, with one or half of our molecules being N15 and the other half being N14. And so if we run these in a centrifuge tube, if we run the solution from these cells in a centrifuge tube, we should end up with a heavier N15 band and a lighter N14 band. So let's talk about what happens in the semi-conservative. In semi-conservative, if we start with our N15 molecule and we conserve half of our molecule in each of the daughter cells, we should end up with a molecule that is actually half and half. That is half old and half new. So we get this sort of N14 and 15 intermediate band. We get a 15, 14 intermediate band. And so I'm going to actually draw two things real quick. One is this new intermediate band is going to show up at sort of a halfway point. This is your 15, 14 combo band. Now, if we run this again, if we run a second round of replication, we're actually going to end up with this result here, where we have this combo band in our centrifuge tube. And then we also have a band that is the N14. Now, this is because in the second round of replication, we actually get some molecules that are only N14, and we never get any N15 bands. With our dispersive method, remember we start with N15, and we end up with these combo molecules that are half N15 and half N14. You get the picture. So no matter how many rounds of replication we do, we always end up with only these combo bands. And so we end up with only that intermediate combo band. We never see a 14 or a 15 band again. And if you continue, let me go back. Sorry, I'm switching back. If we go back to semi-conservative and you continue to run this, you will eventually end up with only that N14 band. You will never end up with only an N14 or N15 band in your dispersive experiment. So let's look at their actual results. So this is another diagram showing if you have your N14 only, you have this light band up at the top. If you have N15 only, you have the heavier band down at the bottom. The LL refers to light, so two light strands. HH refers to two heavy strands. If you have that dispersive model that I was showing you over here, this one if you have that model, you're going to end up with your light and your heavy bands. And if you have any of those combo molecules, you're going to end up with that N15 combo, this intermediate sized band. And so what they found was that they actually got this combo band with the intermediate. So if this band is our N15 band and this band is our N14 band, they found that they actually get, initially in the first generation, they get that intermediate band, which is exactly what we said we would see over here. Sorry, you get this N15 combo band. And then after 2 generations, they get that N14 and the, excuse me, going the wrong way, and the combo band. And so they were able to show that after 1 generation, you get that combo molecule. After 2, you get the combo molecule in your N14. And if we look at data even farther, that intermediate band eventually disappears because you end up with so little of that N15 DNA that you actually don't even see it. And you end up with only the N14 band. And so they were able to show that semi-conservative replication is the process that is actually happening. All right, so this slide is actually sort of a summary. After 1 round, you can rule out conservative because you never see a single N15N band again. And there are never any intermediate molecules, and you wouldn't expect that from the conservative because you never have any intermediate molecules. After several rounds, you start to see less N15 and mainly N14, and you see that intermediate, which you would expect to see for your semi-conservative. Now let's talk about dispersive. Because of the way dispersive worked, you would actually expect to see intermediate bands. But you would, because you're constantly combining old and new molecules, you would actually always continue to see intermediate bands. So we can rule out dispersive because we would never lose the original parental band there.